All right, before I begin, I need everybody just to kind of take a second and lock in, because this is a lot of chemistry. All righty, so today I will be presenting my summer research on lanthanide chlorides and their crystallography. So to begin, before I begin, I'd like to acknowledge my research professor at LSU, Dr. Slava Baranets, and my grad student, Tamira. They both helped me throughout the lab and helped me learn a bunch of lab techniques and how to handle myself around radioactive materials, which is kind of stressful. All right, so everybody knows what this is. This is the periodic table of elements. We kind of use these elements in everyday life: hydrogen, helium, lithium, beryllium, boron, carbon, and oxygen, just to name a few common ones. But I mean, if you look down at this bottom row, these lanthanum and actinium, these two rows are lanthanides and actinides. And they are kind of relegated to the bottom of the periodic table because they're not really commonly used, and many don't think they're that important. However, this is what my research solely focused on. All right, so lanthanides, there's this phenomenon with lanthanides called lanthanide contraction, which means that in the real world, they kind of cluster together, and it's really hard to separate and do research on them. So after you separate them, in order to study them, you have to separate them and then use advanced crystallography techniques. All right, so let's define some things. So we talk about crystals in terms of lattice structure, and this little green sort of structure is one lattice. Now, this blue highlighted region inside of this lattice structure is one unit cell. And then a Bravais lattice structure are the 14 different ways that these lattices can be arranged and found in the common world. All right, so these are the 14 lattices that crystals could possibly be organized in: triclinic, monoclinic, orthorhombic, tetragonal, trigonal, cubic, and hexagonal. And they all have different angle lengths and then side lengths, which have different properties and mean they do different things. And this is important in our research so that we can predict what they may be used for and how applicable they could be in society. All right, so what I did. So there's this inorganic crystal structure database. It houses all of the compounds ever found, all of the inorganic compounds in the entire world. And what I did was I went in the lab, and I sat down on this database, and I was just looking around for compounds that weren't there, So all the lanthanide rare earth metal compounds that weren't there. So eventually, I found two that weren't discovered: thulium, Cl36H2O, and holmium, Cl36H2O, and wanted to solve the crystal structure for those. So then first, we import all of these materials from China, and sometimes they are impure, meaning that they might have carbons or hydrogen added onto them, which means that we can't really do accurate testing on them. So first we have to test for these compounds' purity. Then, using an advanced technique called single crystal X-ray diffractometry, then we can use that to solve the crystal structure and how they might be organized, which then will help us predict their properties. All right. So the first one, powder X-ray diffraction. So first what you do is you take 1 gram of the substance, you grind it up with a mortar and pestle into a fine little powder, and you place it on a glass slide. And you insert that slide into a powder X-ray diffractometer. And what that does is it takes a bunch of X-rays and it shoots it out at this powder, and these X-rays reflect. And based off this reflection, using a law called Bragg's law, it relates X-ray wavelength to atomic distance, and that allows us to kind of determine how pure this compound may be. So if you look to the right, at the bottom, this is a graph of what our expected graph that we should have got, and the top is the graph that we actually got. And each of these little peaks, it represents electromagnetic reflections. And since you see that these peaks are roughly the same in both what we got and what was expected, we can kind of tell that the compound that we had was really pure, which meant that we could do single crystal testing on it. All right, this is the really important technique. It's called single crystal X-ray diffraction. It's done using a $500,000 microscope. There's probably only about 2 in the entire state, which is pretty cool to work with. And what you do is you take this crystal, you cut it with a scalpel into 1 micrometer in length, and then using a microscope, and then you insert that crystal onto this goniometer head, that's a little spiky tube thing, and then you take that head and you insert it onto the microscope. This microscope is cooled using liquid nitrogen. Okay, so what this does, you basically, again, you take this X-ray and you shoot it at this crystal, and then once it hits the crystal, it diffracts, so it scatters. And all these reflections scatter onto this film. So our film that we used, it was some copper backing. So this is an example in the bottom right of our film that we got for one of our compounds. Now, all of these little highlighted regions are reflections. So these are areas where our X-rays scattered, all the light emitted scattered. And since in atoms, all their radiuses are the same. Every single atom has a radius that's identical, and it's held constant. And the distance between two atoms are always held constants, which means that if we can know the distance between two atoms, we can know where they kind of lie and where their atomic position is. So using this Bragg's law again, which relates X-ray wavelength to atomic position, we can kind of tell how far that the light scattered, where atoms are sort of going to be, and then we can use that to eventually solve its crystal structure. So at the top right, this is kind of our raw data once we translated what we got on this film into a program called OLEX2 software. And this is what kind of the initial crystal structure kind of looks like before we standardize it. Our results, so eventually we were able to determine, using this technology, that our two compounds exhibited a monoclinic P2C structure, which means that two of its angles were held constant while one was different. And then in the top right, we can kind of see trends. So our thulium compound and our holmium compound, which are sort of on the right on the little green decrease kind of line, that was kind of sort of what we predicted, which means that many of its properties might align with some of the properties in the other compounds. These are what the crystal structures actually look like. So if you look at, if you find one of them in real life, these are what the compounds sort of look like. On the left is one molecule, and then in the middle is what it looks like in one unit cell when it's kind of expanded, and they're all kind of joined together. And then to the right are what the actual lattice structures sort of look like. All right, a lot of chemistry talk. Why does this matter at all? Okay. So semiconductors are materials found in all electronics and everything that we do. And what they do is conduct electricity. We need them to function. We need them every single day in order to make our things sort of work. Now, there's this process called semiconductor doping, in which we take a bunch of metals and we kind of insert it into these semiconductors to make them more efficient and make them run faster and better. And what we sought to do is maybe we can use these lanthanum, or these lanthanides, these rare earth metals, as semiconductor dopants to potentially make our electronics better and make our lives run a little bit more smoothly. To conclude, I guess, one quote that my professor told me was that I may not be solving cancer. This research really isn't that applicable, but it's contributing to the vast field of science, and everybody can really learn something from that. So go science.